Part of the Washington eScience Institute, we would like to acknowledge the Village Plan, shared watershed, Ms. Amish Talela. We are using both Zoom and in person. So for the folks who are remote, we have a Q&A feature in the Zoom, and please feel free to use that to ask questions. At the end, Adam will answer any of the questions. Same for our speaker today is Adam Glasser. He's joining us as part of our mini seminar series shared with the Engineering Data Science University of Washington. Adam joined the Allen Institute in Neurodynamics in 2021. Works on multi-scale molecular neuroanatomy, focusing on the development of hardware software tools for large-scale imaging projects. Prior to joining the Allen Institute, he worked software research in the where he's now he earned his PhD in engineering science college. Yes, and with that, here to I would say the microphone, Adam, but I would say to the presentation.
Okay, excellent. And you can hear me okay?
Yeah, we can hear you just fine.
Perfect. Okay. Well, thank you for the introduction, and thanks for the opportunity to share some of my a lot of my past work, and I'll talk just a little bit about future work. As was mentioned, I recently moved over to the Allen Institute for Neurodynamics, but I was a postdoc at UW in the mechanical engineering department in John Liu's lab for about 6 years, and that's going to be the majority of what I talk about today. And so I'll be sharing basically a large effort on many of our parts performing large-scale microscopy for a number of different applications. Oops, let's see. Okay, just really quick at the beginning, just a quick conflict of interest statement that some of the hardware and/or software, which I'll share today, is being commercialized by Alpenglow Biosciences based here in Seattle, of which I am a co-founder and shareholder. Okay, so to start with the motivation for everything I'm going to talk about today, it really relies on the fact that current histopathology methods, and I'll go into what I mean by this, rely on really a century-old technology. And so for those of you who might not be familiar with what histopathology is or what the workflow is, it's basically the gold standard for analyzing any tissue which is removed from the human body to study disease. And so the way that this has been done for over a century, and it's still currently done, is a biopsy or a surgical procedure would remove some tissue from the human body. And after that, the tissue has to undergo a number of preparation steps. It's first dehydrated to remove all of the water, and that's so that we can actually embed it in a paraffin wax block. And the reason that it needs to be embedded in wax is because the current state of the art is that we would do essentially 2D imaging of this wax block by sectioning very thin 3 to 5 micron pieces off of the top of the block. And then we would mount that on a glass slide, and then we would stain that. If people are familiar with H&E slides, that would be stained with hematoxylin and eosin. And from the entire wax block, you might end up with a handful of slides, which then ultimately are manually analyzed in most pathology labs using a lab microscope. And so something I really want to drive home about this workflow is that in the end, far less than 1% of the tissue is analyzed. And this little representative image here is actually of an H&E slide with the pink and purple staining. And so there's major implications of the fact that such a small portion of the tissue is actually being analyzed. And some disadvantages of this workflow, if I can go a little bit further, are that it's slow. This entire process takes multiple days. It's slide-based, which inherently limits things to 2 dimensions. It's destructive to the sample. Once you cut it off of the wax block, you've forever removed that tissue from the larger block itself, and it's non-digital. None of the data in this workflow is digital. And as I mentioned before, this is really our gold standard for diagnosing and studying disease. So there's tremendous implications for many different diseases, for example, cardiovascular disease, cancer, respiratory diseases, you name it. And so those were not just a random listing, these are actually the leading causes of death in the US. And so there's tremendous potential to try and imagine a new way of doing this workflow. And let me use an anecdotal example, or I'll kind of show on the screen. And if this was interactive, I would maybe ask and make this slide more interactive, but virtually I'll just have to go through it myself. But I'm gonna start with this 2D image here where I'm just showing randomly 0.1% of the pixels. And I would ask if this was interactive, if anyone could tell me what this is a picture of. And I think you'd be very hard pressed to give me an accurate answer. Even if I up that to 1%, I think it's still very challenging to figure out what exactly that image is of. Now at 10%, I think maybe some of you would be able to tell me what this is an image of. And then with 100% sampling, it becomes clear that this is a picture of the Mona Lisa. And so obviously sampling just 0.1 or 1% or even 10% of, for example, the tissue may not be giving you an accurate picture of what's going on inside, for example, with a disease state. But there's obviously a challenge. Doing 100% sampling poses a huge data and also technological challenge to realize this. And so I'm going to go through for the majority of the talk a lot of my previous research while I was at the University of Washington, which was focused on trying to transform the histopathology workflow from a 2D to a 3D world. And so what we envisioned 6 or 7 years ago is that similar to the current workflow, you would have some surgery or biopsy procedure where tissue is removed from the body, but the preparation would be much different. And actually, the first focus area of my previous research was into so-called tissue clearing and whole mount fluorescent staining methods, and I'll unpack that a little bit more as the talk goes on. Really, the core area of my research was on developing custom optical systems to do high content 3D imaging of these tissues once they're prepared. And then, as is very relevant to this seminar series, and actually, admittedly, may not be the largest portion of this seminar, is on the computational tools for acquiring, storing, and visualizing these data sets. And so the advantages of a 3D pathology workflow are that we have volumetric 3D data. We're doing 100% sampling of the tissue. It's non-destructive to the tissue. And most importantly, in many ways, the data is digital. It's not analog. And so if I go back to the fact that this could be used to diagnose and study disease, it could really improve our ability to do these things. And the primary focus of my work for the last 5 or 6 years was solely looking at cancer. Okay, so in terms of impact, it obviously it seems like at face value, it's better to do 3D imaging, but really biologically or clinically, why would this be useful? Well, one such biological structure would be what I'm going to call convoluted structures. And so in this little cartoon, I'm showing a 2D view, for example, one glass slide that would be prepared using the traditional methods. And if I asked you, what are we looking at? And you see a few cross sections of ellipses and circles, it's very, very difficult. And this is what a clinician or a pathologist would also be looking at. It's very difficult to infer really what's going on in 3 dimensions. But if we could look in 3D, you would see this tree-like structure. For example, you may see that all of the structures come together on the top and bottom of where you're looking. And so you simply cannot tell from a 2D image what's actually happening in 3 dimensions. Similarly, there may be complex cell distributions. Here I'm showing red, green, and blue cells. On a 2D slide, you may think that that blue cell in the center is actually closer to the red cells than the green cells. But again, if you could look in 3D, you may see that actually there's several green cells right above and below where you're looking, and they're very close to the blue cell as well. And finally, another category would just be sparse cells that are very rarely found. And so if you're simply taking 2D views or 2D slices through the tissue, you may never ever see them in 2D. But if you could look in 3 dimensions, you may see that there's actually a few foci of these within the tissue volume. And so these have real biological relevance. Convoluted structures could be vessels, lymphatics, or glands. Complex cell distributions could be the spatial relationships between immune cells. And an excellent example of sparse cells could be rare metastatic cells that are often the initiators of tumors and cancer. So really what we could start to do if we could look in 3D is ask questions such as what is the true status of the cancer? How can we try and treat the cancer better? Why is the treatment not working? Where is the cancer spreading? Things like this. And so there's an excellent review that John Liu at UW and myself and others published, I think it was 2 years ago now, that really goes into a high-level overview of this in much more detail. So when I started my postdoc, we realized that there was a new opportunity to sort of realize this 3D pathology vision that we had. And that's that these so-called optical or tissue clearing techniques were rapidly progressing, and there was a new one coming out every other month. And here I'm listing a few of them with their acronyms. Actually, CE3D was developed at UW by Michael Gerner in the immunology department. But there's many such as Clarity, probably the most famous one, but many others which for one reason or another have DISCO in the acronym, such as UDisco, 3Disco, VDisco. And I won't go into the details, but the long story short is that these chemical protocols allow you to render tissue completely transparent. And as a microscopist or an optical engineer, such as my training, this really changes the game and allows you to acquire three-dimensional information from these tissues in very rapid and robust ways. And so how can we actually capture this rich 3D information? This is not a comprehensive list, but sort of three main categories of ways people have tried to do this. One would be known as knife edge scanning microscopy. This actually operates on the paraffin block that traditional histology uses. And the idea here is that you sort of like a robotic deli slicer, you just brute force cut your way through the tissue. There's disadvantages to this technique. One, it's quite slow, and two, it's still destructive to the tissue. A more common way of doing it would be known as laser scanning microscopy. And so here, if you can see the animation, you raster scan a point optically through the tissue, and you're able to acquire a three-dimensional volume that way. This is non-destructive, and you can do this on these so-called cleared tissues, but the disadvantage is that it's very slow because you're only capturing one pixel at a time. And based on the title of my talk, most of what I'm going to talk about today is on this new form of microscopy known as light sheet microscopy. And the main idea here is that rather than raster scanning a point, what you can do is introduce another objective at 90 degrees to the imaging objective. And if the tissue is very transparent, what you can essentially do is blue beam here creates a sheet of light within the tissue. And if you make that sheet of light at the focus of the imaging objective, you can just take a two-dimensional image and capture millions of pixels all at once. And then you can sort of tile and move your way through the tissue to acquire a 3D volume. The advantage here is that it's also non-destructive, but most importantly, it's extremely fast. And it sort of makes 3D imaging of these tissues feasible within a reasonable timescale, unlike laser scanning microscopes. So to go into a little more detail, what is light sheet microscopy? This is the image from Wikipedia, if you were to go up and look up what is light sheet. And I think this 3D view sort of shows what I was describing on the previous slide a little bit better. You can introduce a sheet of light, which is this dark blue cartoon sort of hourglass here. And that sheet of light you can just place at the focal plane of your imaging objective. And as I mentioned, you can directly capture millions of pixels over a 2D field of view onto a camera. And really, technologically, the only speed limiting factor is how fast can the camera take images. And so most people within the field are currently using these scientific CMOS cameras, which can generate up to 1 gigabyte of data per second. And so you can rapidly capture this information. And so traditionally, light sheet systems were actually not developed for imaging, excised tissues, but really imaging live specimens. And here, if these videos are playing, I think these are some of the most stunning videos that people in sort of the developmental biology field have captured using these types of microscopes. You can do time-lapse imaging over unprecedented timescales without damaging the tissue or the embryo. And this is traditionally how light sheet had been used, say, going back to its inception, maybe 15 or 20 years ago. So when I started my postdoc in the mechanical engineering lab, we had these clearing protocols, and we sort of needed to design a microscope to capture the data. And these light sheet systems come in many different flavors, but if you remember back to what I said 2 slides ago, really all you need is 2 objectives and you need to place them at 90 degrees. And then you can sort of spin that 90 degrees around however you want. But really that's the main design requirement. And so this is the very first light sheet system. This is sort of a top view with a cuvette. This is a implementation 3 years later. Now we're looking at the side. You still have this sort of cuvette design. There's this inverted system where now you have 2 objectives like this. But really none of these were compatible with the large human tissues that we wanted to image for our pathology applications. And so, sort of starting from a biodesign sort of viewpoint, the clinical need was that clinicians really wanted, in this case, a flatbed scanner for tissues. That was really what we were tasked with trying to solve. And so, if we think of a paper scanner, as an engineer, this is sort of what's happening. You have some glass plate, you have a camera underneath it, and you simply can place a piece of paper on top. It's very easy to use. Many of us or all of us have actually done this procedure. It's extremely fast. And so if we take that 90-degree orientation of objectives, this is how it would look. And as I've drawn it here, there's actually some issues. If we go back to our Physics 101 class, if we have these light beams coming from air at some angle into glass and then some other, say, the tissue, the beams are going to refract horribly at this interface, and you're not going to be able to form an image. And so, yeah, this is sort of a cartoon showing a similar effect where if you stuck a pencil in a glass of water, you'd see this sort of discontinuity here, even though that's realistically not what's happening. And so this sort of aberration would prevent this system, as I've drawn it here, from working. So the innovation we came up with early on was that we could actually place a hemisphere of glass right here. And then if you traced along this hemisphere, at any place where it intersects with the 2 light paths, the light would always be traveling normal or perpendicular to the surface of the hemisphere. And this would essentially prevent any refraction from happening. And so if we zoom in a little closer, this is sort of what I'm talking about. And the red arrows are showing that everywhere things are at 90 degrees. And so you don't get any aberrations. And now this sort of new light sheet design could actually work. And it would be very compatible with pathology, easy to use, easy to mount tissues. This is a video of the very first system running. This is actually not a cleared tissue. This is a fresh surgical resection. I'm not going to go into that work for the purposes of this talk, but the principles are the same, that you could also place a cleared tissue on top and perform imaging. And so within a matter of minutes, with the high-speed camera and the light sheet approach, you're able to rapidly acquire imaging data from this entire piece of tissue. And so this is not the same piece of tissue I just showed on the previous video. This is going back to the clearing idea. The very, very first thing we imaged was a prostate biopsy from the UW Medical Center, which was cleared. So you can see on top it's opaque, and after clearing, it's now very transparent. Now, if we took a single traditional histology slide from this specimen, you would get one 2D cut, as I mentioned, and you could zoom in and you could look at the cancer that way. But using our 3D light sheet system, we were able to actually image the entire biopsy in 3 dimensions. And so with this data, sort of tying things back to the clinical relevance, why would this actually even be important? Well, for prostate cancer, the grading is done upon the actual 3D structure of the glands. And normally on a single 2D slide, pathologists will just have to make a grading call based on what they see in 2 dimensions. So we did a small little blinded study with our collaborators, Nick Reeder and Larry True, and we scrambled the images and we showed them one by one, and we had them grade the prostate cancer as a function of depth into this data set. And what's interesting is we saw 2 things. One, they changed their own answer for the grading as a function of depth, and they often did not agree with each other as a function of depth. And this has tremendous implications because this grade of 3+3 means that the patient would go on active surveillance, they would go home and they would come back for biopsies a year later. Whereas if the grade was 3+4, they would immediately undergo surgery to have their entire prostate removed. And so this is actually tremendously important clinically to make this call correctly. And what we noticed was after visualizing the entire Z-stack video as I'm showing it on the screen right now, both pathologists agreed that the Gleason pattern should be 3+3 in this case. And so this was really encouraging early evidence for us that there could be tremendous value in pursuing this technology. We got great press as well. It was nice that the NIH director actually acknowledged our work and was also excited about this. And so we pushed forward based on clinical feedback to design a new and improved microscope. So in collaboration with this company in Portland, we essentially improved the optics of the system. So if the first system was providing roughly 2 by 7 micron resolution, we pushed this new system to below 1 micron and about 3.5 to 4 microns in the axial Z dimension. And we also improved how thick of a tissue we could image on this second system. The first system could really only image very thin tissues, and this new system could go up to 5 millimeters thick. So this is a view of the second system. This is now a video that's going to show how it operated. And we really ramped up the prostate biopsy imaging application. And so here you can actually see a 3D printed chip where we've placed a dozen biopsies that are all cleared and fluorescently labeled. And now the system is acquiring data from them one by one. And there's actually 2 dyes, which is why you see the green and red laser going. And in a completely automated way over an entire overnight run, this system can in an automated way image all 12 biopsies, generating terabytes and terabytes of data. And the video will just play, and you can see that it goes through each of the biopsies and it finds the bounds automatically. And so after an overnight run, this is the data that the system would produce. These are all 12 biopsies. And if we zoom in on a single one, this is a zoomed in view of biopsy number 8. And I'm showing 3 different orthogonal cut planes through the data to really demonstrate that this is true 3-dimensional data all the way through the thickness of the biopsy. And if we zoom in further, in all 3 dimensions, you can see that you can clearly resolve, in this case, the green label is the nuclei of the cells within the biopsy, and they're lining these glands, which is forming these rings. And I mentioned that that's what's clinically used for grading the cancer. And so you can see that we have well-resolved images in all 3 dimensions. And if I now show this sort of pseudo-colored pink and purple data set from the new system as a function of depth, I think you can appreciate that we definitely did improve the resolution with this system. And we met our goal of essentially providing imaging data which could be approached, although not being quite as comparable as to what's typically seen on a glass slide by a pathologist. But this is approaching the level of detail that they would need to be confident in grading the cancer. And so, part of the reason that the system that I showed, we had this 3D printed chip and we were able to mount 12 biopsies all at once was really because we needed to do this in a very high throughput manner. We were collecting tons and tons of data, and what we were working towards and actually have recently completed prior to me moving to the Allen Institute was a well-powered Department of Defense funded clinical study in John Liu's lab in collaboration with the pathology department at the University of Washington. And so in a retrospective study, we actually got biopsies from a biobank of tissues for patients. And for each patient, we imaged 6 biopsies. And what we were able to do over the course of several years is actually image 200 patients worth of biopsies. That was about 1,200 biopsies, 1,200 hours of imaging, and over a petabyte of raw data. And really what we wanted to do through this study is acquire all of the 3D information from these biopsies and then compare back to the 2D pathology and see with this biobank of tissues, is 3D information better at predicting the ultimate patient outcome than if you had done things in 2D? And I should have updated this slide. A graduate student in John Liu's lab, Weisa, actually published a study that really went into, say, the machine learning and the image analysis, which was performed on this data to actually try and quantify how much better 3D is from 2D. And so if you're interested in looking into that paper, I'd be happy to point you to Weisa's paper after the talk. But this system, so everything I've described was really laser focused on prostate cancer, but I do want to mention that this system had applications that we explored outside of human tissues, outside of cancer. And so there's also use cases in immunology. And so in this case, we took mouse organs from Michael Gerner's lab in immunology, and we mounted them on the system. We performed clearing. And then we're also able to image these mouse organs in 3 dimensions. In this case, 8 lymph nodes from this mouse, as well as the heart and also the lung. And with fluorescent staining, you're actually able to selectively highlight the, I believe, endothelial cells along the bronchial tree of the lung. And also applications in kidney research. This was in collaboration with Josh Vaughn's lab in the chemistry department. And so this is a form of tissue clearing, but a little bit different in that during the clearing process, you can also actually physically swell the tissue to become larger, so-called expansion microscopy. And so Josh is really a world expert in this area. And what you're able to do is enlarge the tissue by, for example, a factor of 4. And so even though optically everything's remaining the same, you effectively improve the resolution of the imaging system by however large you're able to expand the tissue by. And so in this case, a factor of 4 gets the effective resolution of this type of microscope down to just a couple hundred nanometers. And so the video on the right is showing a small snippet of this very large slice of kidney, which we imaged in 4 different colors that highlighted different biological structures within the kidney. Okay, so about 2 years ago into my postdoc, I decided, what was going to be the next step for this type of imaging device. And it really boiled down to the fact that to tackle many different applications, something our previous imaging systems could not do was what I'm going to call multi-scale imaging, or specifically hierarchical multi-scale imaging. And so what I'm showing here on the turret is in a pathology lab, what a pathologist or many researchers would like to do is to be able to rapidly switch magnifications to see a large field of view and then zoom in when they found an interesting area. And this is something that none of our previous microscopes could do. And so on the bottom, you're seeing an example histology slide where someone is zooming in and zooming out while they switch objectives. And so in 3 dimensions, there's a tremendous need for some sort of hierarchical multi-scale strategy. In 2 dimensions, the data size and the time it takes to acquire the image scales with the resolution squared, which is already quite painful. If you double the resolution, things become 4 times more painful. But now in 3D, things scale with the cube of resolution. And so it's quite imperative that you start to think about these sort of multi-scale strategies for the imaging experiments, depending on what the user's needs are or what the scientific questions needs are. And so really what you could do with multi-scale imaging is you could acquire the entire volume at some coarser resolution, which is sufficient to identify an interesting region, such as this green square here, and then only image that green cube at the highest resolution. I won't go into the details of the hardware too much, but if you look at the center plot, essentially what we were able to do on this newest system is we had 2 imaging modes. One was quite coarse in resolution, about 4 microns and 4 microns, so isotropic data. And then we could actually go even higher resolution than any of the previous systems. And so we were able to achieve below 500 nanometer resolution in the X and Y direction and approaching 2 microns in the Z direction. And all while doing this, we also pushed the maximum specimen thickness that the system could accommodate even higher. And so we went from less than 1 millimeter to 5 millimeters. And now in this newest iteration, 1 centimeter thick tissues can be imaged. And so this really now allows us to start imaging very large, thick human tissues for pathology applications, as well as entire intact mouse organs, for example, mouse brains, for neuroscience or preclinical applications. This is a view of the system that is on the left. Again, I won't go into the hardware too much for the purposes of this seminar series, but we have made the entire hardware design for this system open, and it's already being reproduced in several institutions across the world. And it's also being, as I mentioned, commercialized by our local Seattle startup company, Alpenglow. So this is the type of imaging data the newest system can provide. I guess as a teaser, because I came to the Allen Institute, focused heavily on the mouse brain for this particular paper. But this is an example of a mouse brain where the arterial vasculature is labeled with an antibody. And then in the cyan channel, you have the nuclei. And so this is the entire brain imaged quite quickly at the lower 4-micron resolution mode. And you can appreciate that you can resolve things with detail all the way through the volume of the brain. And now if we zoom in, this is sort of that 4-micron resolution. It's difficult to see, but now if we switch to the high resolution mode, you can very, very clearly even see subnuclear details. And you would easily be able to segment individual cells and nuclei to do quantification. This is another brain imaged actually with my new team here at the Allen Institute, Jayaram Chandrasekhar and Karel Svoboda. This brain has sparse labeling of neurons within the brain. And so you can see with the low resolution mode, we're able to zoom in and see individual axons and neurites. And for example, here is one particular neuron. And now if we zoom in and image that neuron with the high resolution mode or other regions, we can capture much, much more detail, but on localized volumes and not over the entire brain. And this is a Z-Stacks where if you zoom in, you can see individual varicosities along the axons themselves. And to kind of, again, drive home the high throughput, large data scale of these types of experiments, about a year and a half ago now, we initiated for this study an example of studying brain metastases in mice. And this was sort of an international collaboration to give you an idea of how these types of experiments can really come together. So ourselves at the University of Washington had built this microscope. Collaborators at the Riken Institute in Japan were experts in the tissue clearing and labeling procedures. And then a team at the University of Tokyo had the mouse lines and the cancer cell lines to sort of prepare the brains themselves. And so at the University of Tokyo, the brains were prepared and those tissues were delivered to Riken, where the clearing and labeling was performed. And then those samples were FedExed to us in Seattle to do the actual imaging. And then once we imaged the tissues, we could share the data back over the cloud. For example, we put the data on AWS. And so the timeline of how this came together was in October, the project was initiated. And then just 1 month later, we had done all the experimental procedures. And the results were that we had imaged 6 entire mouse brains. We had imaged 50 metastatic regions of interest at high resolution and generated about 40 terabytes of data. And so this is an example of one of those brains. The cancer cells and the metastatic growth is shown in cyan. And then there's a nuclear counterstain in magenta, which you see throughout the rest of the brain. And so you can appreciate how sparse and localized these metastases are. And with this sort of hierarchical multi-scale imaging approach, we can actually identify certain regions of interest centered on the metastases and only image those regions at higher resolution. And so in this particular brain, we didn't image all of the metastases, but I think we imaged around 10. And now if we zoom in on one of these metastases, you can appreciate that the higher resolution data is able to resolve a lot more detail than the lower resolution mode. And now if we look at a Z-stack, we can actually see very, very fine detail of the biology of these metastases. And it's captured in an extremely data and time efficient manner. And so within one brain, we can capture multiple regions of interest. And then in a single imaging session, 6 brains and up to 40, I think, regions of interest in this case. And finally, I think everything I've shown, nothing has been quantitative, but in a collaboration with DR Vision, now part of Leica in Bellevue, we actually did segment out the lumens and the cancer cells in this data. And this is results from their software Avia, showing them in there, the cyan is the metastatic lumen of sort of where did the metastases grow, and the nuclei are shown in magenta. And so we can do some quantification of different cancer cell lines. In this case, the cell line MDA231 has a lower volume to surface area metric, which is sort of a pseudo metric for how round or spherical the growth patterns are. And it's different from this OSRC2 cancer cell line. And also, we could look at things like what is the cancer cell density in the vessel co-option regions versus inside the metastatic colonies. And so you can really start to use this type of data to do quantification of the biology. Okay, now in the the last 10 or 20 minutes, I would like to finally get back to some data science, some actual data. Now that I've given an overview of sort of how we captured the data, why we captured the data, what our motivations were. I hope this will be interesting, and I'll go into a little bit more of what does the data look like and what are some considerations for large-scale 3D microscopy data. So when we acquire these data sets, we obviously have this three-dimensional array of pixels. And for example, along X, Y, and Z, there may be some size to the data set. And this is per channel. So we have one data cube for channel 1. And then each voxel within this data cube can be 8, 16, 32, or 64 bits. And typically for microscopy, the cameras that are used produce 16-bit data. But now if we have another channel, we have a whole another cube of data for channel 2, and we might have a third channel. And so we end up with many of these imaging volumes that all should have the same size, and they should ideally all be co-registered to one another so that the colors all overlap properly. And I won't go into the details, but actually the chromatic alignment of channels can be tricky, and that's an entire potential algorithm and field in and of itself. And something which is very, very commonly done in microscopy and probably other fields with imaging data is an efficient multi-scale representation of the data. And so here I'm showing the native resolution of the data, which may be, say, n by n by n for the 3 dimensions. And to load that entire data set, say, into RAM or to visualize it on a computer screen, if this is 100 terabytes, there's no way that you could possibly do that. And so what is typically done is that the data formats which are used in our field, you will store downscaled versions of the data. And so, for example, you would store a 2x downscaled version, a 4x downscaled version, so on and so forth. And so depending on how zoomed in you are on the data when you're visualizing it, you only load on demand what's necessary. And if you go through the math, this actually doesn't add too much data to the overall size of your data set. And if you kind of expanded out this math to even 16 downsampled versions of the data, it can only add about 14% data. And really how this is used visually, I hope this 3D cartoon is somewhat clear, but imagine if this was in 3D and your viewpoint was where this sort of sun-like sphere looks. You're closest to this top right corner. And so you want to render that at higher resolution than areas of the data set which appear to be farther from where you are. And so this is going back to it can be much more complicated than just loading one whole downscaled version, but you can actually have this sort of hybrid approach where depending on your visualization software, you are on demand loading different resolutions of different parts of the data set. Another really important factor in this type of data is selecting the order of axes. So for my particular data sets, we'll restrict it to just X, Y, and Z for the 3-dimensional data, but you could have a time component. Actually, as I've already mentioned, there's a color component. So it's more than 3D, but for the purposes of this slide, let's assume we just have X, Y, and Z axes. So when we store the data on disk, we have to choose an ordering to the axes. And so in this case, if we choose an X, Y, Z order, what's going to happen on disk in the memory is we would have the blue would be location 0 and then 1 in memory, and then Y is the next dimension order. So we would move up and this would be location 2, 3. And then if we moved back essentially in Z, we would have 4, 5, 6, and 7. And so the memory layout in a linearized way would be 0, 1, 2, 3, 4, 5, 6, 7, and it would be corresponding to the actual data set in the way I'm showing it here. But you could imagine another scheme where you choose ZYX ordering. So now the order of sort of bits as they go to memory would go 0 and now 1 back in Z, then we would come up to 2, 3, 4, 5, 6, 7. And so although in memory, it's still just linearized, depending on how we choose the order of axes, you would have access to the data in much different ways. And so this is a very important consideration where you may choose the ordering based on how you think you may process the data and read it back in to make that step the most efficient step possible. Along these same lines, another very important factor for these types of imaging data sets is actually chunking. And what do I mean by this? Well, if we had the three-dimensional volume, you can actually break it up into chunks. And so here, what I'm showing is you essentially have a bunch of these 2D planes in X and Y, and every Z step is a different chunk. And actually, a good way to think about this is if you just had, say, TIFF files or JPEG files, you had simple images, they would be essentially chunked in this manner, where you would have one image per plane. So the chunk size in Z is just 1. So the chunk size here, I'm going to say, is 10 by 10 by 1. Now, the implications of this are that if you want to read anything from, say, the 4th Z plane, you have to read through all of the pixels in the initial chunks to get there. And so this is potentially, depending on what parts of the data you think you might access, a very inefficient way of doing things. Instead, you may be able to use isotropic chunking. And so here I'm showing the same data set, but chunked 5 by 5 by 5. And so depending on, if I just read all of chunk 1 into memory, I now have access to, I have access in X, Y, and Z, all in a sort of uniform manner up to a certain point before I'd have to read in chunk 2. And so the memory layout kind of abstracted away from individual pixels, but now the chunks would be 1, 2, 3, and 4. And reading all of chunk 1 means very different things in this case versus this case in terms of what you're able to readily access. And this can also have tremendous implications for compression as you start to think about ways to compress the data. Obviously, being able to compress with three-dimensional context for the information within the images is much more important than, for example, just having an entire 2D plane. And for a lot of my work moving forward and some of the prior work, it really helps to kind of contextualize what does a large-scale imaging experiment mean and what are the data pipelines that need to support it going to have to support. And so for many traditional microscopy imaging experiments, on the y-axis, I'm showing how fast is the data coming in off the system. And on the x-axis, what is the total data set size? And so for many experiments, there may be 100, 200 gigabytes, and you're only acquiring the data at tens of megabytes per second. Now, I think for a lot of what I just showed at the University of Washington work, we pushed this quite a bit, and we were acquiring individual data sets, which were on the order of a terabyte, and we were acquiring the data at hundreds of megabytes per second using these light sheet systems. And a lot of what I'm going to be pursuing in my future work at the Allen Institute is scaling this even further to individual data sets, which are on the order of 100 terabytes, and acquiring them even a little bit faster using more state-of-the-art cameras, in some cases 2 cameras, and so now even higher data rates. And so this isn't the entire story. So here I'm showing these 2 axes, which are very important considerations. But there's actually another axis which is important for my future work, and I think needs to be considered. And that's the cadence of how often are you planning on acquiring these data sets? And will it be 1 per year? Will it be 1 per week, or will it be 1 per day? And so if we now think of this third axis, again, many researchers may be only acquiring a handful of data sets annually, maybe 1 per month. And for a lot of my UW work, we sort of acquired these large data sets very quickly, but we only acquired them a couple of times per year. And really another sort of crux of the work which I'll be pursuing at the Allen Institute is that we are hoping to pursue or acquire these types of data sets every few days. And so in terms of engineering, not just the hardware, but the software and the data science aspects of keeping up with this deluge of data, it's going to be a huge component of my future work here. And so I don't have any actual data or anything to show, but just one final slide on sort of what is my future work. It's sort of taking these 3D imaging methods and, as I mentioned, scaling them up to pursue this new area within neuroscience and doing very large scale imaging. And in this case, not looking at cancer, but trying to contribute to a very large goal, which is much bigger than myself. I have no background in neuroscience, but I have excellent team members around me here. And there's also institutions all across the globe pursuing this larger goal of trying to understand the brain and understand behavior. And ultimately, if we can understand that, how we can improve our ability to treat, prevent, or even cure different disorders of the brain. And so this is actually the type of imaging experiments which we will be doing. This really builds off of a prior project that Jayaram Chandrasekhar, who's leading the anatomy effort here of the team I've joined. This is the mouse light project, where individual neurons were mapped within the mouse brain. And when he was running this project at Janelia, they were able to map thousands of individual neurons. But in order to do this tracing, the imaging has to be done across the entire brain at high resolution. And so that entire multi-scale imaging approach that I mentioned previously to sort of only image select regions of interest would not apply here. This requires brute force high resolution imaging of the entire large volume, which is why the data sets become so large. And with that, on my last slide, I just want to say that if anyone is interested in learning more about the Allen Institute for Neural Dynamics, I would love to chat further, and feel free to reach out. And also, if anyone is interested in a career potentially at the Allen Institute for Neural Dynamics, there's many positions coming online, particularly in the sort of data science and software engineering space, given the scale of the data sets that we'll be acquiring. And with that, I just want to thank everyone who's supported this work. It's really been an excellent experience within Seattle, within the University of Washington in particular, as well as collaborators around the country, as well as the globe. And thank you so much for your time, and I'd be happy to take any questions.
Thank you, Adam. Can you hear me okay, Adam?
Yes, yeah, I can hear you.
Okay, great. Thank you very much. I love the visuals. It's absolutely stunning. I have two quick questions for you. The first one is, can you tell me or tell us what the difference is between the Allen Institute for Brain Science and the Allen Institute for Neural Dynamics?
Yes. I guess I might not actually be able to give the, it's evolving with time, but they are completely separate institutes. And the Brain Science Institute is in a transitional phase. So portions of the Brain Science Institute, such as Mindscope and others, will continue. But really, the Allen Institute for Neural Dynamics is born out of Karel Svoboda's vision. He's the new director here. And we're focused on really trying to link all of the anatomy work I'm showing, as well as other modalities, to behavior. And so really trying to do this sort of holistic approach to neuroscience. But I can't give a much better answer than that. I think Karel or others would do much better than me.
No, that's super. It helps me sort of place it in the context and understand the more transitional aspect of this, or translational, excuse me. My second thing is thinking about, if you go down to atoms or you go up to galaxies, imaging in both of those spaces has benefited from not recording the raw images, but recording catalogs. So the positions and shapes and locations of objects is obviously a lot less dense, especially when you have so much sparsity. And maybe it's just when I'm looking at your images, you've conveniently sort of blacked out all those sparse regions. Is there any possibility or potential utility to convert these into catalogs of objects versus the raw image data itself?
Definitely. So the pathology application is a little bit more challenging, although because in those cases, at least with the sort of pink and purple stains I was showing for H&E, sort of everything is lit up. And so, but ultimately, if you could segment out the nuclei, then just store their XYZ position and maybe some other metadata about shape and sphericity, things like that. But your comment or your question about cataloging for these new neuroscience applications is spot on. So it's very sparse, and essentially the first step, once the data is captured and sort of stitched together, is to segment out the neurons. And I think it went by very quickly, but on like one of the last slides, you might have seen this sort of like tracing of the neurons. And so once that step is done accurately, then the entire 100 terabytes ultimately collapses down to a graph of the neurons, which may be less than a megabyte in size. But getting through that entire pipeline, especially the segmentation part, currently requires a lot of manual proofreading, which is actually the rate limiting step for these types of experiments.
I see it naively. You might think that segmentation is parallel, but I suspect between frames and between regions of space, segmentation is not. There are actually connections. We have a question from the audience here.
Hi, Adam. Super cool stuff. I had a question about how often we need that sub-micron resolution diagnosis, or the visuals that you've got there. Is that leading to new abilities to diagnose things? Because that seems not even comparable to what we've been able to get manually using microscopes.
Yeah, that's a great question. I think it's tricky to answer. I think what is necessary ultimately, because things scale to the third power when you push resolution higher, as low as you can get away with, I'd say. But a lot of our work in designing the hardware was focused on sort of achieving a similar level of detail or resolution as what a pathologist would see when they use their standard lab microscope to look at a slide. And for doing any sort of grading and not diagnosis, I have to be clear there, but just any grading to sort of get an idea of what's going on, they stated that they would typically use a 20x objective. And that's sort of how we designed that second system I showed to reach that level of detail. But there are select cases where they would go to a 40 or a 60x objective to start to look at subnuclear features. And so it's tricky to try and fully replace the current pathology practice. I think where I see the 3D pathology fitting in is sort of an additional adjacent test. It would never replace the sort of way things are done now, but it could be an added test where you could look in 3 dimensions if something seemed ambiguous and you wanted a more definitive answer.
Okay, we have one question from the Q&A on Zoom. It's from John Liu. Great talk as always, Adam. Consider the sparsity of neurons. Is there any interest in the Allen Institutes in techniques like light field microscopy that have the advantages for sparsely labeled objects?
That is a great question. I think we're not pursuing it now. It's not on the plan, although John is absolutely right that some of these other techniques like light field could have advantages here. I think the biggest thing that is going to drive is driving our work at the Allen Institute is to get really isotropic resolution. I didn't go into that too much, but these axons get down to 100 nanometers in size, and they can pass within 100 nanometers of one another. And so those are the ambiguous points for segmentation algorithms of you don't know which one went which way. And even with the best optical approaches, we will never be able to do the imaging at 100 nanometer isotropic resolution. But using some of our light sheet techniques, we hope to say achieve 300 to 400 nanometer isotropic resolution. And so with these light field techniques, they may suffer a bit and not be able to achieve that. But it isn't something I've dug into too much. And so it could be a really promising way to go about trying to do this.
Great. Well, thank you very. Oh, we have one more. Evan.
Hey, thanks for kind of hearing me. Yeah, thanks for coming and talking. So I can see how like the amount of data for these 3D images is so explosive, and I can also appreciate how the multi-scale imaging can reduce that. But it sort of seems like making that jump, you're going from something that's just a brute force scan to something that requires some sort of like you got to know where you want to zoom in on. So I guess how in like an overnight process can you send in like a biopsy, have it imaged, and have an like is there some sort of algorithm that determines which areas it wants to focus in on? Or
yeah, I think you hit the nail on the head that everything I showed with those metastatic brains was me manually setting up each region of interest by looking at the lower resolution data set. But absolutely, you would ideally want to have some sort of closed loop, algorithm, either with if it's simple enough, just simple image processing to sort of pull out where you see any signal, if the labeling is good enough, and then only generate higher resolution scans on those areas. Or it could be much more complicated. For example, if it's the sort of H&E data where you have stain everywhere, then you would need to put it through some sort of learning algorithm that was trained with some gold standard truth to identify what regions to zoom in on. And since John is on the webinar, I know, I believe John is working on these exact sort of things, but it's a great question. Yeah.
Gotcha. Thank you. Yeah, that's where my head was going to with the learning.
Super. All right. Well, I think that's it. Let's thank our speaker one more time.
Thank you so much.
Wonderful. Thank you very much, Adam. This was a wonderful fit for our program. And with that, we're going to close the formal portion of our seminar.
Awesome. Thank you so much.